My name is James Gagnon. I just started my PhD in the laboratory in Sherbrooke, in Quebec, Canada. So today I am going to present the newest work on Mesoplasmophorum and where our laboratory is directed with it. So, yeah. So just to make sure we all talking about Mycoplasma microarrays, or more interested in Mesoplasmophorum that's just near Mycoplasma. So it's a molecule. And what makes Mesoplasmophorum particularly interesting is its small genome, around 800 kilobases. It's non-pathogenic nature. It grows well under standard conditions with a building time of only 30 minutes approximately, which makes it safe and easy to work in the lab. So to begin, I'd like to briefly walk you through the workflow that we made up with Mesoplasmophorum. We've already performed high density transposon mutagenesis, or HDTM, but they have already been presented by Gagnon, the one that mostly made all the computational analysis that I will present today in the third minimal cell workshop. So if you're interested, and the HDTM will give us a layer to identify which genes are dispensable or none in different selective media. So, but today I will just talk to you mostly about the RNA-seq that we've made in the laboratory, and again with my project afterward of the building of a synthetic genome of Mesoplasmophorum. So first of all, this heat map shows the RNA-seq expression profile of a Mesoplasmophorum gene across different sugar condition. We compared the transcriptome in the undefined ATCC 1161 medium that it's not defined. Yeah, I just said it. And in a semi-defined CMRL-based medium supplemented with various sugar, including sucrose, fructose, glucose, maltose, creolose, and mannose. For today's presentation, I will not focus on the difference between ATCC and CMRL medium, but if you are interested, Anthony Duval, an ancient member of the laboratory, presented about it on the third minimal cell workshop too, so you can pick it there. So what's interesting about that is the most sugar condition in the CMRL medium produce with a relatively similar expression profile. However, when mannose is used as the energy source, we observe a dramatic shift in gene expression. This we can see that with the volcano plot here, with CMRL with the 6 sugar compared to mannose, and we see really big shift in the gene expression, and with the glucose compared to mannose. If you want another comparison with this pairwise analysis, we can see the comparison between glucose and fructose, and we see that it's really a difference. Really, it's very the not same expression profile that we see with mannose. The genes upregulated in CMRL6 sugar were mostly related to energy production and growth, such as ribosomal protein and ATP synthase components, whereas the genes induced in mannose were enriched in glycolysis and carbohydrate catabolysis pathways. Mannose appears to be a poor energy source in this organism, possibly due to the catabolism through promiscuous enzymatic pathways, and mannose may be diverted to anabolic routes that maybe need a further explanation to do for us. But together we want to just keep the mannose for now away from the other source of energy to compare the energy source in a bigger picture. Because if we look at the correlation between mannose and all of the other energy source tested, we can see that it's really clearly different again. And with the PCA, we can clearly see that the CMRL with glucose, fructose, sucrose, and trehalose and maltose are clearly in this spot right there. And we can have ATCC 1161 with the 6 sugar right there, and the CMRL with the mannose only with those replicates at the right left. Okay. So now the problem with the pairwise analysis is that we always need to compare to one condition, which tend to miss the bigger picture of what the sugars are with the expression profiles and everything. So we tried to find another way to analyze our data. We came up with a clustering approach. It's not me that's made it. It's Simotano in our laboratory. So I will try to explain it the best that I can. Yeah, so just switch slide. Okay, so we came up with that to help better visualize what is going on with all the sugar. It's a UMAP, a dimensionality reduction method that help us group genes based on how they vary across a condition. Each point here represents a gene, and those that behave similarly tend to cluster together. We then use 22 distinct gene groups that gave us a clearer picture of the response that we have in the genome. And we can identify modules of genes that might be co-regulated or functionally linked. Now you may ask, why did we choose 22 groups of genes? Sorry. For the rest of the presentation, we will be sticking with this group of genes because it will be too long if we look at every genes here. So if we're back with the 22 cluster, we look at two metrics, the silhouette scores and the Davies-Bouldin index. Ideally, a high silhouette score and a low DBI indicates well-separated and compact clusters. However, gene expression data is noisy and overlapping, so perfect separation is really unrealistic. So instead of aiming for mathematically optimality of these clusters, we prioritize biological interpretability. So with 22 clusters, we achieve a practical balance, enough resolution to detect meaningful genes module without over-fragmenting the data. So it's a functional choice that reflects the complexity of the system more than strict genomics separation. So if we go back with the cluster that I talked to you later, this one, I represented it in a Cleveland graphs. So we see the 5 genes that are represented in this cluster. And we see that instead of the pairwise analysis, we have a more global image of this. And we see that genes PEG 212 and 13 are the 2 genes most expressed in fructose. And we clearly see this too with the track up right there with the RNA-seq signal. For this Cleveland graph, an important detail is when the marker is a circle, so like that, it means that the fold change is considered robust and the replicates are tight and the difference from the mean is reliable. But when it's an X, like this, that means that the fold change might still be large, but it's less statistically supported. So it doesn't mean the effect isn't real, just that it's harder to say with confidence that it's consistent across replicates. So basically, this is a measure of the fold change robustness. Yeah, so with the comparison, with the pairwise analysis that we've made to just be sure that our clustering approach is reliable, we can see with this volcano plot here of glucose compared to fructose that these 2 genes here are the PEG 212 and 213. So we clearly see the same results of the pairwise analysis, but with a bigger picture. So after that, we look at the genes that seems to have a bigger expression in different sugar, and we've made that for all the important genes that we saw. And if we stick to the PEG 212 and 213, We can see the annotation from different sources. So we got RAS, KEGG, RefSec, GenBank 14, and Patrick. And those genes are described to be hypothetical or nothing, as we see here. But for the 213, we can see that it's either glucose-specific PTS or N-acetyl glucosamine transporter or just a generic PTS subunit. And there is no clear consensus, and this makes it difficult to confidently assign a function, especially when we're trying to link it to a specific sugar response like fructose, because we clearly see that it was more expressed in fructose and not in glucose, like we can see here in orange. So this kind of ambiguity is common in minimal genome work, and it reminds us that annotations are helpful, but they are not always definitive, especially for genes with overlapping or context-dependent function. That's why combining RNA-seq data with clustering and condition-specific analysis is important to help us go beyond static labels and actually understand how genes behave. So with that, it closed the little apartheid for RNA-seq. And I will dig more into my project, that is to build synthetic mesoplasmopheron genome. So I present about it in the Toy Minimal Cell workshop. I said there that I will be done 1 year after that, but finally no. But I will explain you what we've been through. So the plan here is to take the genome of Mesoplasmophorum to devise in 10 sectors of like 90 kb. And for the genomic fragments, we separated in 12 PCR fragments that we amplified. We make a vector with a centromere for the yeast and 2 oxytrophic marker. We will then transform it in a Saccharomyces cerevisiae C31, showing that all the oxytrophic marker are really went away with the deletion procedure. And after the transformation, we will get rid of URAT3 selection marker to make sure that we not accumulate them over all the transformation. We will do the extraction sequencing, and after we will cut at the extremity of the vector with not one enzyme that it's not present into the genome of Mesoplasmophorum to have linear fragment after that. We will do the same thing for the 10 sectors that we will co-transform with, again, the vector of centromere and leucine at with URAT32 with for oxytrophic marker. And we will add the tetracycline resistance marker gene. You will understand later why. After, like I said, we will transform it again into Saccharomyces cerevisiae. We will again get rid of the URAT3 marker because it's not needed after that. We will do extraction, sequencing, and after be able to have the synthetic genome. Right now, for the progress, we have 2 sectors that have been made. The 8 others seem to be more difficult to assemble in the yeast. So I don't know really what's going on right now, but maybe with this talk we have some idea. We after that take the genome that we assemble. We will do a transplantation of genome into Mycoplasma capitulum that we already know that it's able to have this genome into the cells, 2 genome at a time into Mycoplasma capitulum. And with the tetracycline resistant marker gene, we will do the selection with tetracycline and be able to boot up Mesoplasmophorum synthetic genome. Like I said, I started working with the yeast like 2.5 years ago, and we have an idea after that that we can use phages to use to boot up synthetic transcription that we can be able to put in the capsid. But after like 1 year of effort with that, we decided to abandon this strategy and came back with the yeast. So this is why it's not really advanced compared to the last time. So with that, we will still need to establish the procedure to build Mesoplasmophorum synthetic genome with only PCR fragments. So it's really less costly with just 2 steps of assembly into the yeast, so it will be faster. It will challenge our understanding of genome design. We will publish RNA-seq result, to publish additional results for the TNC that we are right now writing the article. And after that, using the RNA-seq, HDTM, the iGEM models that we already made in the lab, and the GCVI minimal cell, we will try to build minimal version using all of these data. So with that, I'd like to thank my teams, my director, and thanks to Labo Caras that helped me later on with the genes transformation. I see that Bogomil and Daniel are in the talk, so thanks again for you.
Jeremy, if you were to look at those genes with the way you're analyzing this, you still are looking at all genes under two different conditions. But another way to do this would be to say that you standardize genes relative to, say, 16S ribosomal RNA transcription as a standard, and so you're looking at differential expression of genes under two conditions. I suppose you can do it either way. Can you comment on that?
Yeah, we try to do that too, but it's not with just two conditions. We look at all the sugar that we talked about it, and we try to look too with the ATCC with sugar and no sugar, and we try to look at too with CSY. And so yeah, we got a lot of condition, and we try a different approach to look at the, like you said, the RNA-seq signal. But yes, could be an approach to look forward. Yeah.